very small correction to the title, because I initially suggested exploring the late Miocene diatomaceous event in the Mediterranean, but actually I realized that probably the best title would be exploring the late Miocene diatomaceous events in the Mediterranean. I am convinced that there were more than one single diatomaceous event that occurred during the late Miocene in the Mediterranean region, and I will try to convince you. By the way, this is a very brief framework of the presentations that I will give today, some of the major points that I will try to touch during my presentation. And well, let's start with a very brief introduction with a question, which is the Mediterranean actually a good place for studying marine diatomaceous sediments? Well, if we consider the relatively low diatom productivity in the Mediterranean water column, which is mirrored by a recent sedimentary record, which is mostly characterized by the abundance of calcareous deposits, the first answer to this question would be probably no. But what about the geologic past? Actually, if we consider the geologic past of the Mediterranean region, and especially the Neogene record, sedimentary record, for sake of simplicity, comprised between 23 and 2.6 million years ago, well, the story is changing a lot because in particular, the Messinian stage, which is the uppermost stage of the Miocene, which is comprised between 7.2 and 5.3 million years ago, was very diatom rich. So if we consider this time interval, well, the Mediterranean is definitely a good place for studying the marine diatomaceous sediments. What about the Mediterranean during the Messinian? Well, it is very important to note that since 7 million years ago, so with the onset of the Messinian, the 7.2 million years ago marks the onset of the Messinian stage, there was a significant basin restriction. And well, you can see from this map that at about 7.2 million years ago, there was a restriction of one of the major getaways that connected the Mediterranean basin with the Atlantic Ocean. And this restriction progressed up to 6 million years ago with an almost definitive closure of the Mediterranean to Atlantic gateway that was responsible for the onset of the dramatic paleoenvironmental change that occurred in the Mediterranean basin, which is known as the Messinian salinity crisis. During the Messinian, in the Mediterranean, there was a very important influence of riverine runoff, which was much higher than present day, 3 to 4 times stronger than present day. This is mostly because of the strong contribution of the African continental interior. And these had very important implications for the water column structure of this restricted basin, especially because it influenced the stratification of the water column. You must imagine a huge amount of fresh water reaching a restricted basin. And, of course, this led to the stratification of the water column with very important consequences on the water column oxygen levels, for example, and therefore on the biogenic sedimentary record. The sea surface variability, sea surface temperature variability was very high. The onset of the Messinian is characterized by a cooling trend with a subsequent rebound at about 6 million years ago. There were very important changes in the carbonate factories of the Mediterranean during the Messinian, during the early Messinian, so between 7 and 6 million years ago. There was a widespread occurrence of coral reef complexes. I'm not an expert of these carbonate factories of the Mediterranean, but I strongly suggest to take a look to this paper of Irene Cornacchia on Earth Science Reviews. This coral reef expansion has been attributed to the cooling climate, to the cooling of the climate that occurred during the early Messinian. And between 6 and 7 million years ago, there was a very relevant increase of the siliceous deposition. You can see from this map that all the Mediterranean was involved in this important increase of the diatomaceous deposition. The restriction progressed, and this led, as I told you before, to the evaporitic deposition with the Messinian salinity crisis at about 6 to 5.3 million years ago, when the Mediterranean was literally turned into the youngest salt giant of Earth history. A huge amount of evaporitic sediments accumulated in the Mediterranean, more than 1 million cubic kilometers of evaporites, mostly gypsum and halite, accumulated everywhere in the Mediterranean basin. So, resuming, the Messinian sedimentary record of the Mediterranean is an excellent laboratory for investigating, for understanding the climatic oceanographic perturbations that occurred in this restricted semi-enclosed basin during the Neogene, during the Upper Miocene. Let's start discussing the lower Miocene diatomaceous accumulation. So the first diatomaceous event that occurred before the onset of the Messinian salinity crisis between 7 and 6 million years ago. This diatomaceous accumulation was characterized by this cyclicity. Okay, we can appreciate in some cases in spectacular outcrops this cyclic stacking pattern consisting in the alternation of diatomites, sapropels, so organic rich deposits, and marly sediments, so a mixture of calcareous and terrigenous sediments. This cyclic stacking pattern has been attributed to an astronomic control, and in particular to the precessional control. So for sake of simplicity, let's say that during precession minima and insolation maxima for our hemisphere, there was a huge amount of fresh water that reached the Mediterranean basin, especially, as I told you before, from the African continental interior. And this led to the stratification of the water column with the consequent weakening of deep water formation and the establishment of anoxic conditions in the deep waters, which favored the accumulation of organic rich sediments, the so-called sapropels. While during precession maxima, insolation minima, a drier climate characterized the peri-Mediterranean region, and this led to enhanced mixing of the water column with oxygen conditions partially replenished during the deposition of the diatomaceous deposits and the marly sediments. What are the main interpretations that have been provided for explaining the origin of this dramatic increase of the diatomaceous accumulation in the Mediterranean? There are many interpretations, to be honest, but the most important ones, at least those more widespread in the literature, suggest a link with upwelling currents, so the rise of nutrient-rich cold waters along the continental margins that are able to enhance the concentration of nutrients in the upper water column. And this interpretation, for those of you working with diatoms, is mostly based on the occurrence of Thalassionema nichoides in these deposits, which is actually an opportunistic diatom that may thrive well in many different environmental conditions, to be honest. I think that there is a sort of, let's say, the application of Thalassionema nichoides as a universal proxy of upwelling is, in my opinion, overemphasized, especially in the Mediterranean region. But by the way, the other hypothesis suggests a linkage with bottom water anoxia. So let's say a sort of a Black Sea scenery with the establishment of anoxic conditions that favored the preservation of biogenic silica, according to the authors that support these hypotheses. Anyway, I think that we are limited in the interpretation of these diatomaceous accumulations, basically because we lack in-depth biosedimentological studies of these sediments. The most majority of the studies concerning the diatomaceous deposits of the Mediterranean are based on smear slide preparation, basically, so typical micropaleontological approaches, which are absolutely useful, but, on my opinion, should be coupled to other approaches, less invasive approaches, non-destructive approaches that may help in better understanding the composition of these sediments. So the first case study is from the northern Mediterranean area, from the Piedmont Basin. I don't want to make you bored with geological frameworks and stratigraphic frameworks. By the way, for sake of simplicity, just consider that the lower Messinian diatomaceous deposits occur along with these stratigraphic successions, which are characterized by the alternation of marly deposits and muddy organic rich deposits that toward the top are locally enriched in opal. And these are, in my opinion, the fantastic diatomaceous deposits of the Piedmont Basin. You can appreciate this fine lamination, which consists in the alternation of pale and dark laminae, often interrupted by coarse grained layers, bioturbations, phosphate nodules. There are also some microfolds, which may be suggestive of the instability of the sedimentary environment of these deposits. The paleontological content of these deposits, it's astonishing, both if we consider both the macro-paleontological content, so the fishes, especially fossil fishes, are very abundant in these deposits. There are also some plant remains. In some cases, even insects have been found, fossilized. But, of course, for most of you, the most interesting part is related to the micropaleontological aspects. So we have both plantic, benthic diatoms, but also foraminifera, plantic, benthic, calcareous nanofossils, and an overlooked, in my opinion, and not completely understood rich assemblage of other silicifiers, such as, for example, sponge spicules, silicoflagellates, hebridians, chrysophyte cysts, and endoskeletal dinoflagellates, or even radiolarians. So a very rich and heterogeneous assemblage, which is suggestive of very high productivity in the water column as well as on the seafloor, probably. Well, but my focus is not exactly the micropaleontology in this presentation. I would like to show you the high potential of laminar scale petrographic observation of these sediments. How it works? Well, it's not so complicated, I would say. We sample these diatomaceous deposits, and then we split the samples into different fractions that are employed for polished thin section preparation by means of epoxy resin, and we do not treat a fraction which is used for the preparation of lamina fragments that are observed with the SEM in order to implement the observations made on the polished thin sections. Well, the result is a description of the, let's say, basic units of these laminated sediments, so the lamina, and their organization at a higher, let's say, hierarchical level. So their organization in packets, in bundles of packets, etc. By the way, I just want to highlight that in this research, there are, on my opinion, a lot of fantastic papers that have been published, but there are two very important references, in my opinion. One is the paper by Chang, Grimm, and White of the 1998, published on Palaios, about the Miocene Monterey Formation. And the other one is a collection of papers by Hallenkamp, 1999, Paleoclimatology and Paleoceanography from laminated sediments. They are excellent source of information for those interested in exploring the fantastic world of the laminated diatomaceous deposits. Anyway, I would like to show you now just some results of these investigations that we did on these diatomaceous deposits from the northern Mediterranean. By means of the laminar scale petrography, we were able to distinguish different lamina types. And for example, the heterogeneous biogenic lamina, the main components of these lamina, of course, are the silt and clay-sized grains, as well as calcareous nanofossils. There is a consistent amount of calcareous nanofossils in the matrix, and benthic diatoms, mostly inshore diatoms. And one curious fact is that by means of this kind of approach, you can better appreciate some diatoms which, on average, by smear slide preparation, are very difficult to be preserved because you crush them, you destroy them. For example, arachnoid discus, or in this case, in panel I, some isthmia. So these allow, in my opinion, a higher degree. This allows a higher degree of preservation of some big specimens that, on average, are broken during the preparation of smear slides and therefore destructive approaches. How to interpret these lamina? Well, we suggest that they were related to a humid seasonal climate during which the primary productivity in the basin was dominated by calcareous nanoplankton. And, well, humid conditions favored clastic and biogenic inshore input into the basin. The second lamina type, which is, on my opinion, very relevant for better understanding the origin of the diatomaceous deposits in the Mediterranean, is represented by the so-called diatomaceous mud lamina. Well, these lamina are typified by a concentration of large centric diatoms, mostly of the genus Cosinodiscus, as well as in the matrix the presence of calcareous nanofossils. How to interpret them? Well, we suggest that they reflect the fast deposition of diatoms that were adapted to exploit stratified waters during the warm season, forming the so-called deep chlorophyll maximum. This is basically a shade flora that was able to grow under stratified conditions in the darker portion of the water column. And well, the massive sedimentation of these diatoms may have occurred with the onset of cooling and therefore the breakdown of the stratification. This mechanism has been described by Alan Kemp in the 2000. I strongly recommend to take a look to this paper for the interpretation of these kind of assemblages in the laminated sediments. And finally, the diatomaceous ooze laminae. And these are also very intriguing. They consist in the massive accumulation of pennate plantic diatoms, needle-shaped, mostly Thalassionema. They are closely entangled, and well, the laminae are characterized by a very good preservation of the lamina boundaries, a very good preservation of the lateral continuity. And how to interpret them? Well, in this case, we suggested a relationship with the cold season and the onset of mixing conditions in the water column that were able to reintroduce the nutrients that were stored in the deeper part of the water column during the stratification. And this favored the massive proliferation of these diatoms and the consequent nutrient exhaustion, the aggregation, and the massive sinking of these diatoms. Again, this is a mechanism which is, on my opinion, very overlooked in the study of the diatomaceous deposits in the Mediterranean region, but which was described, for example, by Grimm in the 1997 and named self-sedimentation, so the massive sedimentation of diatoms at the end of bloom episodes. This approach allows the reconstruction of small-scale sedimentary cycles, which are, for example, the seasonal sedimentary cycles. So these sediments have a great potential for understanding small-scale sedimentary processes. In the most majority of the cases in the geologic archive, it's impossible to reconstruct these processes. In the case of the diatomaceous deposits of the Mediterranean, I think that we are in front of a very high potential for such kind of reconstructions. The second case study is from the central Mediterranean. We are still in the stage of the lower Messinian, so before the onset of the Messinian salinity crisis. We are in the Abruzzo region. The paleogeographic, geologic, and stratigraphic setting is completely different. We focused on this portion of the stratigraphic section of Capo di Fiume, which consists in this alternation of diatomaceous layers and muddy, organic-rich deposits. These are some pictures from the field. You can appreciate even in this case, the fine lamination of these sediments, sometimes interrupted by coarse-grained layers. And there are also very big bioturbations in these deposits, which are quite interesting for the interpretation of the oxygen level at the seafloor during their deposition. But by the way, these deposits are also full of fossil fishes. You can see on your right just a part of the very huge amount of fossils which are preserved in these sediments. But well, the focus is on this polished slab of diatomite, which is characterized by, as I told you, by this fine lamination. But the laminae are organized also in lamina sets, so mud-dominated packets, which are probably indicative of a high grade of detrital input for prolonged time, and therefore this led to the dilution of the biogenic content of these sediments. and diatom-dominated packets, which are pale, which are characterized by probably were originated by a drier climate that favored the proliferation of diatoms and the low dilution of the biogenic content of these deposits. These are, in my opinion, astonishing diatomaceous deposits in the Mediterranean. Anyway, what I want to stress is that even in this case, there is a fundamental component of these diatomaceous deposits, these very large centric diatoms that are key components, especially of the best preserved lamina in these sediments. So these raise some questions, in my opinion, coming back to the main interpretations that have been provided for the interpretation of these deposits. Was upwelling the main controlling factor of the accumulation of these deposits? Well, probably, but not necessarily, and especially not the only triggering factor. Thalassionium onychoides is, as I suggested at the beginning of this presentation, an opportunistic diatom, which is not necessarily an indicator of upwelling. We must consider the key role that was played also by diatoms, which are adapted to exploit completely different water column structures, the stratified water columns especially. And we must consider the potential role played by enhanced runoff in favoring the stratification of the water column during the diatomaceous accumulation. And another point is related to the benthic component. I'm not an expert of benthic diatoms. I can just show you how abundant they can be in some lamina types. They can provide a lot of information potentially. So this is an open question, in my opinion, that must be better addressed, the benthic assemblage of these diatomaceous deposits. And the second point is related to the anoxic conditions that have been considered as a very important controlling factor for the preservation of the lamination, for example, of these deposits. We must consider that there are some specific mechanisms of diatom aggregation and sinking that may enhance the preservation of the laminated fabric of these rocks, even under oxygenated conditions. So the simplistic association between laminated fabric and anoxic conditions is not enough, in my very humble opinion, for suggesting that anoxia was the main controlling factor for the preservation of these sediments. So more research are, of course, needed, but I think that coupling destructive approaches with less invasive approaches, such as lamina scale petrographic observation of these sediments, may be very useful for better understanding the origin of these sediments. The second part of my presentation is about the diatomaceous record of the Messinian salinity crisis. So we are dealing, with the second part of the Messinian, the second half of the Messinian, during the Messinian salinity crisis between 6 and 5.3 million years ago. During the Messinian salinity crisis, there was an apparent dramatic drop of the biogenic sedimentation, especially the diatomaceous one. And you can see from this map, which is quite obvious, that there is a strong difference between the early Messinian diatomaceous deposits in blue and late Messinian diatomaceous deposits in red. So there is an apparent reduction of the diatomaceous accumulation. And the classic interpretation that was provided for explaining this apparent opal crash is that the evaporation of the Mediterranean basin related to the closure of the Atlantic to Mediterranean gateways led to hypersaline conditions in the water column, and this led to the drop of the marine biological productivity with dramatic consequences on the Mediterranean marine biota. Is this a fact or a fiction? Or in other words, does the search for traces of life, and especially diatoms, in the sediments that formed when the Mediterranean was turned into a giant salina worth the efforts? Does it make sense to take a look to the Messinian sediments formed during the Messinian salinity crisis looking for diatoms or other paleobiological content? In my opinion, the answer is yes. This is the third case study. We come back to the northern Mediterranean area. This is the stratigraphic setting, completely different. We were dealing before with the sediments formed below this red line, which marks the onset of the Messinian salinity crisis. We are considering the sedimentary successions above this red line, which are characterized by the presence of gypsum, of course, and muddy organic-rich shales. And this is the section that we are considering, the Castagnito section in the Piedmont Basin. It is characterized by this cyclic stacking pattern of consisting in the alternation of gypsum-rich beds and dolomitic mudstone beds. And toward the top of the section, it is possible to record these diatom-bearing sediments. What if we take a look to these sediments with a non-invasive, let's say, approach? So looking for for example, the content of biogenic silica and its preservation. If we consider the diatomaceous shales, where they are characterized by the highest abundance and best preservation of biogenic silica, which intriguingly is characterized by is consisting in these mats, very well preserved, of thalassionema, thalassiotrix, and rhizosolenia. So diatoms, which are not suggestive of hypersaline conditions in the water column during the formation of these deposits, which is quite interesting in the light of the interpretation of the Messinian salinity crisis onset as related to hypersaline conditions. By the way, the second lithology that we are considering now is represented by the diatom-bearing mudstones, which are characterized by common but very altered diatom remains, which are characterized, you can see here, by the growing of clays all around the valves, and by the presence of framboidal pyrite, which is a sulfide indicative of bacterial sulfate reduction. So anaerobic degradation of organic matter under mostly anoxic conditions. And finally, the dolomitic mudstones. So the most majority of the stratigraphic section, consisting in these very abundant dolomite grains, bright in the backscatter, you can see here, and surrounded by these flaky clays that we interpret as autogenic, so not related to the detrital input in the basin, but formed in the sediment. For example, by means of the combination of dissolved silica with pore water cations. And in these sediments, there are very rare and strongly altered diatomaceous remains. Even in this case, there is a high abundance of framboidal pyrite. So together with the presence of dolomite, these are good indication of the bacterial sulfate reduction. Why also dolomite is a good indication of bacterial sulfate reduction, because sulfate-reducing bacteria are able to increase the alkalinity of pore waters, leading to the formation of magnesium carbonates, such as calcium magnesium carbonates, such as dolomite, for example. And there are good morphological evidences of these processes, such as the presence of these hollow dolomite. You must imagine the increase of alkalinity around the bacterial cell, which is able to favor the precipitation of dolomite, self-entombing, therefore, auto-entombing the bacterial cell. So the result is a no-low morphology of the dolomite grain. And what about the mineralogy? Very briefly, of course, the dolomite-rich mudstones are characterized by the highest concentration of dolomite, and the diatomaceous shales by the lowest one. So we have two end members, let's say, if we consider the dolomite as a good proxy of the bacterial sulfate reduction. Well, this is a good indication that diatomaceous shales did not experience a significant bacterial sulfate reduction during their formation. And well, this is just for the clay addicted among you. I don't think there are so much clay addicted among you, but by the way, if you are interested about the clay composition of these deposits, especially the amorphous clays, which are the poorly crystalline clays, which are closely associated to dolomite, well, these are the names, so halite, smectite, and vermiculite. Anyway, much more interesting is the biomarker content, the biomarker inventory of these sediments. In the case of the diatomaceous shales and diatom-bearing mudstones, we can appreciate very well-preserved algal lipids, while in the case of the dolomitic mudstones, there are keto holes that we interpret as reflecting the degradation of diols. So they are indicative of the degradation of algal lipids during the formation of the dolomitic mudstones. And the point is, who played a fundamental role in this organic matter degradation? We suggest, according to the biomarker content of these deposits, again, the intervention, fundamental intervention of sulfate-reducing bacteria under anoxic conditions. So what is the main take-home message? What is the interpretation of these petrographic observations combined with geochemical investigation, etc.? That there was, on our opinion, a persistent diatomaceous productivity and sedimentation during the Messinian salinity crisis, but contrasting bottom water conditions led to different patterns of organic matter degradation. So prolonged anoxic conditions during the formation of dolomitic mudstones favored high rates of bacterial sulfate reduction. As I told you before, this is a metabolic pathway which is able to increase the alkalinity of pore waters, leading potentially to extensive biogenic silica dissolution. If so, the consequent buildup of dissolved silica in combination with pore water cations may have led to the extensive clay neoformation that we appreciate in these dolomitic mudstones. So, in other words, the autogenic clays that we observe in this lithology are a sort of proxy of very high-grade biosiliceous productivity that was involved in high grades of degradation by means of sulfate-reducing bacteria. And when anoxia was locally, temporarily interrupted, so this occurred likely rarely during the Messinian salinity crisis. According to what I showed you before, the dolomitic mudstones are overabundant if compared to the diatomaceous shales. And we suggest that when anoxia was interrupted, mostly aerobic degradation of organic matter occurred, with a minor impact on biogenic silica preservation. By the way, what about now, what about the evaporites? So we discussed the diatomaceous component, the diatomaceous deposits that are interbedded locally to gypsum layers. But what about gypsum? This is the point. Does it make sense to look inside gypsum, which is considered as a barren lithology without a paleobiological content? Why? Because basically it should reflect very harsh environmental conditions that were lethal for the most majority of life in the water column or on the seafloor. And this is the fourth and last case study that I present. This is from different Mediterranean localities, mostly in the Western Mediterranean. This is the impressive, very nice ridge of the Vena del Gesso basin in northern Italy. It consists in this alternation of thick gypsum beds and thin euxinic shale layers. I don't want to list all the techniques that we employed for the investigation of these gypsum crystals. We employed ACM, light microscopy, confocal laser scanning microscope. So a lot of different approaches, multidisciplinary approaches, let's say. And we focused on the first stage of the Messinian salinity crisis, which is comprised between 6 and 5.6 million years ago. Okay, and this is the typical, this is the reconstruction of the wall stratigraphic succession in the Vena del Gesso basin. So this one, let's say. Anyway, what are the results of these strange kind of investigations that we performed? Well, there is an extraordinary preservation of biogenic silica in these gypsum crystals. The biogenic silica mostly consists in nano-sized thalassiosiroid and other plantic and benthic diatoms. In some cases, it is also possible to appreciate perfectly preserved specimens of the genus Lampriscus, which, to the best of my knowledge, is very, very rare in the fossil record. They are perfectly preserved in the gypsum crystals. And moreover, another important fact is that not only biogenic silica, but also organic matter associated to diatoms is in some cases excellently preserved in gypsum crystals. Here you can see some threads that can be putative chitin threads associated to thalassiosiroids. But what I think is very relevant to stress is the presence of putative cell ultrastructures in these diatom frustules, which are much likely lipid droplets associated to pigments. So photosynthetic pigments, carotenoids, and chlorophyll. So this is, in my opinion, quite interesting, considering that these diatoms have an age of more or less 6 million years. But beside excellent cases of preservation, there are also clear evidences of biogenic silica alteration and organic matter alteration related potentially to the intervention, again, of sulfate-reducing bacteria. We found framboidal pyrite, we found dolomite, and if you remember what I told you before, these are autogenic minerals associated to the activity of sulfate-reducing bacteria, and we found again the typical byproduct of the early diagenesis of biogenic silica, which are the autogenic clays again. So it's a very complicated scenery, in my opinion, But resuming what we can say, I would say that, well, maybe we are in front of a diagenetic bias that affected the diatomaceous record during the Messinian salinity crisis, rather than an opal crash that was associated to the establishment of harsh conditions in the water column of the Mediterranean area. More research, even in this case, is absolutely needed, but answering to the question that I raised before, looking for the diatomaceous record of this Messinian salinity crisis, absolutely, it worth the effort, I think. So concluding, just a very brief note about the global perspective that, in my opinion, we must consider when dealing with the diatomaceous accumulation in the Mediterranean area during the late Miocene. And to be honest, probably generally speaking, during the whole Miocene, by the way. The Messinian diatomaceous events that occurred in this semi-enclosed basin occurred during the phase of significant increase of processes related more or less directly with the biogeochemical cycle of silicon. And these important ecological transitions occurred both on lands as well as in the oceans. These led to the widespread so-called biogenic bloom of the late Miocene early Pliocene. So my question is, and I have finished my presentation, were the Mediterranean diatomaceous events possibly the local expression of a much wider phenomenon? With this question, I finish my presentation. I thank you very much for your attention, and these are some acknowledgements to my group. So Francesco Della Pier, Giorgio Carnevale, Marcello Natalicchio, Rocco Gennari, Francesca Lozzar, Enrico Nallino, and Nicolo Zanellato, and also to my supervisor when I was in Japan during my PhD, Rick Jordan, and excellent guys of his research group, especially Kenta Abe. Thank you very much for your attention. And if you have questions, of course, I am absolutely available.
Thank you, Luca, for a really amazing presentation. I'm just fascinated by this incredibly dramatic event that took place in relatively recent history and relatively recent geologic history. I don't know, yeah, I've just been captured by it, and you're so soft-spoken about the importance of this work, of what you've done to put it together. So thank you so much.
Thank you, Sarah.
If anyone has any questions, feel free to raise your virtual hand or put your question in the chat.
Yeah, it's probably better also for me, so I can better understand.
I also shared some of the citations that you were speaking of in the chat with people, so that they can have some reference to more.
Yeah, if you need a reference list, I can send you.
Yeah, I wanted to ask Luca about your last statement about this. Is this a local expression versus a global event that we're just not seeing in the other record? And one of the things is this, it seems like such an unusual depositional setting to have this relatively isolated, very much isolated basin that doesn't have a lot of circulation and can doesn't have big ocean currents and doesn't have deep upwelling. Is that part of it? I'm not familiar with the setting of the Monterey Formation. You implied that it had similar kinds of maybe not a preservation, but at least of layer, stratigraphy.
Well, I think that comparison with the Monterey Formation is probably well. We must discuss. We must discuss about similar sedimentary products, but in different oceanographic settings. This is the point. The region where Monterey Formation accumulated is very different from the Mediterranean one. Upwelling was absolutely a very important controlling factor. But I am not sure that this is the best analog for interpreting the oceanography of the Mediterranean region during the Messinian, especially because during the Messinian, the Mediterranean was characterized by a restriction, and as you said before, by a weakened oceanic circulation. So I'm not sure that the Miocene Monterey Formation is a good analog for the Messinian diatomaceous deposition in the Mediterranean area. What is very interesting, maybe for a diatomist, is the occurrence of these Thalassionema nichoides, which is also abundant in the Miocene Monterey Formation, of course, and may be related actually to upwelling currents. So these cold water rise along the continental margin of western United States. But I think that in the Mediterranean, the abundance of Thalassionema nichoides is potentially related to other controlling factors. It's an opportunistic diatom. So if we consider that it may thrive very well also in other environmental conditions, well, the attribution of this diatom to upwelling, it's weak, on my very humble opinion, in the Mediterranean area. In contrast, the high abundance of these centric, very large diatoms, which may be potentially associated to the stratification of the water column, may, in my opinion, be it's actually an overlooked component of the Messinian sediments, but may have a great potential for explaining the accumulation of these deposits in the Mediterranean, which was, as I tried to show at the beginning of this presentation, which was extremely influenced by riverine runoff during the late Miocene. So this may have led to the stratification of the water column, and therefore the proliferation of these deep chlorophyll maximum diatoms, which may account for a very important role for the primary productivity in the Mediterranean during the late Miocene. But of course, this is an open question, in my opinion. Much more researches are needed for deciphering this unusual sedimentary event in the Mediterranean.
Yeah, Rafaela says, fantastic work. I have a lot of points to clarify and explore further. I have to metabolize after reading the publications. Thalassionema, I think, Nichoides and Rhizosaline. Maybe you'd like to speak it yourself, Rafaela.
Yeah, no, because there some analogs are with some results on Antarctic continental slope and area in which upwelling systems are active now, but even during the Pleistocene in that area. So you link the great centric diatoms as stratified water columns, but I think even Rhizosolenia species and pennant like Thalassiotrix even could be linked to stratification and to shifts, even daily shifts of mats in the water column. So we need to have.
We found good evidences of Rhizosolenia mats in the, especially during the Messinian salinity crisis, which is interesting in my opinion, because we may suggest that with the progression of the Mediterranean restriction, the relative influence of riverine runoff enhanced the water column stratification. And these may have favored diatoms such as, for example, Rhizosolenia, which is also able to establish some relationship with nitrogen fixing bacteria. It's a very interesting diatom. So
we have to find analogs, modern analogs in somewhere.
It's very difficult to find modern analogs because you must look for a very unusual setting. It's like it's the big question of the Messinian salinity crisis, for example. It's very difficult to find a good analog of a salt giant because today the accumulation of evaporites at a huge scale, such as in the Mediterranean during the Messinian, it's very difficult to be found in everywhere in the world. And so it's quite complicated when you are dealing with these huge sedimentary events to find good analogs. It's not an excuse, of course. We must look for them, absolutely. But it's not easy at all,
no,
my opinion.
No, okay.
But to get back to your, I think the way you began your presentation is the modern day Mediterranean is not productive. It's, you know, compared to these historical events.
Yeah,
Can you go over that? Why is it now not productive given what it was pre Messinian?
Well, this is a very good question. And the reason for the present day low productivity of the Mediterranean is that it is considered as an oligotrophic basin with a circulation that reduce, let's say, the concentration of nutrients in the Mediterranean waters. So there is a circulation that favor the, let's say, removal of nutrients toward the Atlantic Ocean. And well, this is not the best place, of course, for siliceous primary producers. The other reason could be related to anthropogenic effects, which may have altered the silicon biogeochemical cycle. So the strong increase of phosphate and nitrate compared to silicon, dissolved silica in the Mediterranean waters may alter the assemblages of phytoplankton and leading potentially to the reduction of the diatom abundance in the Mediterranean waters. Of course, I am not an expert of present-day Mediterranean ecology, but well, this is a possible explanation. So the change of the Mediterranean circulation during the Messinian may have played a role in the increasing rate of diatomaceous sediment accumulation. But on my opinion, besides the progression toward the Messinian salinity crisis, there is something more, which is associated to the possible transitions that occurred in terrestrial ecosystems during the late Miocene in the peri-Mediterranean region. There was an expansion of open habitats dominated by grasses, and grasslands are full of silica that may have potentially enhanced the concentration of dissolved silica in the ocean. This is a whole hypothesis, but in my opinion, deserve to be considered for the interpretation of the diatomaceous spike that occurred in the late Miocene in the Mediterranean, as well as in other areas, because the grassland expansion occurred at the worldwide scale during the late Miocene. And another factor that may have been very important was the expansion of deserts. During the late Miocene in the Saharan area, we have the first evidences, the first strong evidences of strong aridification processes. And we know that deserts may play a very important role in increasing the concentration of nutrients, transporting a lot of nutrient-rich material, dust, basically, in the ocean, so favoring the proliferation of phytoplankton. Another hypothesis that was raised at the beginning of the research on the Messinian salinity, on the Messinian diatomaceous accumulation, is a possible linkage with volcanic activity. But there are not so strong evidences of a close association between diatomaceous deposition and volcanic activity during at least the lower Messinian, the early Messinian, sorry, in the Mediterranean area.
We should talk further. There may be some of those in the western U.S. in the Lahontan Basin. Volcanic activity in the, when...
Are you talking about lacustrine deposits?
Yes, I am.
Yeah, okay, yeah. Okay. Yeah. So fresh water.
Leila, go ahead. We'll take your question.
Yeah, thank you very much. Thank you very much for your talk. It's really great for the geologic way. I just one remark. I don't know. There was a theory because I was curious about your layer with gypsum rich with diatom. There is Georges Busson who said that gypsum and halite might deposit under water, not in evaporitic situation. So I don't know if this theory for you is possible. And just I was curious for the upper part of the Miocene, which is very rich either in diatom,
absolutely,
and sponge spicule, and as well carbonate, foraminifera, etc. Normally we have either calcareous lamination and or the siliceous one. So how do you explain this richness, and all is preserved, well preserved, either calcite and silicate? Thank you very much.
Thank you very much, Leila. I am not sure that I understood correctly your question. So are you talking about lower Miocene deposits?
The lower Miocene, it was just for the gypsum, when you showed us the gypsum rich with diatom, which is curious. Yeah.
Yeah, how to explain that excellent preservation of diatoms in gypsum? Well, there's not just a perfect case of preservation in gypsum of diatoms, but also of strong alteration. Well, the point is that we should think about gypsum. We must think about a mineral which is able to, with high grades of precipitation. So gypsum is able to entrap microorganisms very, very rapidly, potentially, preserving them perfectly, because it is a sort of sarcophagus, let's say, okay? There are very interesting papers of Kathleen Benison about the high potential of evaporites, for example, for preserving potential biosignatures on Mars. Just to give you an idea of the possible application of the search for life in evaporites. On the other hand, what we observed in the Messinian gypsum is that there are also evidences of strong alteration of biogenic silica. So it's a contrasting scenery. You have a fast rate of gypsum growing, and at the same time, when gypsum was temporarily interrupted, maybe because of seasonal increase of humidity, fast rate of diagenetic alteration of the material that was subsequently entrapped again in gypsum. So unfortunately, I did not show you the morphology of these gypsum crystals. They are, for example, the massive selenite is characterized by these very fine lamination consisting in gypsum-rich and gypsum and organic matter and clay-rich lamina. And well, this is a good indication of seasonal processes, probably, that during which gypsum deposition was enhanced or briefly interrupted. So we must think about a scenery characterized by very fast environmental changes when gypsum was actively growing on the seafloor, because in these cases we are dealing with bottom ground gypsum. Okay, there are different gypsum lithofascias, there are different kind of gypsum crystals that formed during the Messinian salinity crisis, but this is another story. And
yeah, my point is, if it was evaporitic condition, we would have a metasaline species. That's what we didn't see. That's why maybe it wasn't really dry. And the theory that some halite and gypsum might deposit under column of water, maybe so. So this is for this period, and for the upper period of the Miocene, which is very rich in oil. I was curious about just the preservation, the well preservation of both silicified biogenic diatom, etc., and the calcareous one, the coccolite and foraminifera, etc. This is what I didn't understand. Maybe it's not the same level, or I don't know.
Well, unfortunately,
well, I'm gonna have to go. Sorry. I'm going to the field, so I'm gonna have to end.
Okay.
Leila and Luca, please continue your discussions. And Leila, also you should see the publications. Yeah. Get those out of the chat of Luca's on that amazing gypsum preservation. Okay, and so, thanks very much, because it was great in terms of geological point of view, and the Messinian is a big problem for a lot of geologists. Thank you.
Thank you very much. Thank you very much, Leila.
Yeah, thank you everyone for joining. It's great to see you all, and please have courage in these times and continue to do good work. Thanks so much.
Thank you very much.
Take care.
Thank you, Sarah.
Bye, everyone.